I'm going to read from page 162, Structure of Skeletal Muscles. Skeletal muscle is composed of several kinds of tissue. These include muscle cells, also called muscle fibers themselves, nerve tissue, blood, and various types of connective tissue. Figure 8.1 displays the relationship between muscle and the various connective tissues. Individual muscles are separated from each other and held in position by connective tissues called fascia. 3 separate layers of connective tissue exist in skeletal muscle. The outermost layer that surrounds the entire muscle is called the epimysium. As we move inward from the epimysium, connective tissue called the perimysium surrounds individual bundles of muscle fibers. These individual bundles of muscle fibers are called fascicles. Each muscle fiber within the fascicle is surrounded by connective tissue called endomysium. Just below the endomysium and surrounding each muscle fiber is another layer of protective tissue called the basal lamina, or basement membrane. Despite their unique shape, muscle fibers contain most of the same organelles that are present in other cells. That is, they contain mitochondria, lysosomes, and so on. However, unlike most other cells in the body, muscle cells are multinucleated, contain numerous nuclei. One of the most distinctive features of the microscopic appearance of skeletal muscles is their striated appearance. These stripes are produced by alternating light and dark bands that appear across the length of the fiber. Dark bands contain the primary muscle contractile proteins and will be discussed in detail later. Each individual muscle fiber is a thin, elongated cylinder that generally extends the length of the muscle. The cell membrane surrounding the muscle fiber cell is called the sarcolemma. Located above the sarcolemma and below the basal lamina are the group of cells, precursor cells called satellite cells. Satellite cells are undifferentiated cells that play a key role in muscle growth and repair. For example, satellite cells can contribute to muscle growth during strength training by dividing and contributing nuclei to existing muscle fibers. Increasing the number of nuclei within muscle fibers enhances the fiber's ability to synthesize proteins and therefore assist in muscle growth. In addition of nuclei to growing muscle fibers is a strategy used by the fiber to maintain a constant ratio of cell volume per nucleus. The volume of cytoplasm surrounding an individual nucleus is termed by myonuclear domain. The biological significance of myonuclear domain is that a single nucleus can sustain the necessary gene expression, production of proteins, only for a limited area of cell volume. Therefore, to maintain a constant myonuclear domain, new nuclei obtained from satellite cells are incorporated into skeletal muscle fiber during growth. It follows that to maintain a stable myonuclear domain, nuclei are most lost from fibers. One moment, please. Music's good, but gotta go. Domain nuclei are lost from fibers when a reduction in muscle fiber size occurs, muscle atrophy. Beneath the sarcolemma lies the sarcoplasm, also called the cytoplasm, which contains the cellular proteins, organelles, and myofibrils. Myofibrils are the numerous thread-like structures that contain the contractile proteins. In general, myofibrils are composed of 2 major types of protein filaments. Thick filaments composed of protein, your myosin, and thin filaments composed primarily of protein and actin. The arrangement of these 2 protein filaments gives skeletal muscle its striated appearance. Located on the actin molecule itself are 2 additional proteins, troponin and tropomyosin. These proteins make up only a small portion of the muscle, but they play an important role in the regulation of the contractile process. Myofibrils can be further subdivided into individual segments called sarcomeres. Sarcomeres are divided from each other by a thin sheet of structural proteins called a Z-line or Z-disc. Myosin filaments are located primarily within the dark portion of the sarcomere, which is called an A-band, while actin filaments occur principally in the light regions of the sarcomere called I-band. In the center of the sarcomere is a portion of the myosin filament with no overlap of the actin. This is the H-zone. Within the sarcoplasm of muscle is a network of membranous channels that surrounds each myofibril. These channels are called the sarcoplasmic reticulum and are storage sites for calcium. Remember from your previous physiology courses, the calcium released from the sarcoplasmic reticulum plays an important role in triggering muscular contraction. Another set of membranous channels called the transverse tubules extends from the sarcolemma into the muscle fiber and passes completely through the fiber. These transverse tubules pass between two enlarged protein portions of the sarcoplasmic reticulum called the terminal cistern. All these parts serve a function in muscular contraction and are discussed in detail later in this chapter. Cool, so that's page 162. Structure of skeletal muscle, going to 164. See ya.